very good morning, class. Today we are meeting for a Science and Technology Part 2 lecture. Chapter number 2, that is Living Organisms, Life Processes in Living Organisms Part 1. We had already started and almost we are going to complete the chapter today. But before we move ahead with the chapter, let us first revise and see what we have learned in the earlier classes. In the earlier lectures, we learned about mitosis cell division. What is mitosis cell division? Mitosis cell division takes place in somatic cells. Somatic cells are present everywhere inside your body except your reproductive organs. Reproductive organs carry germ cells. Therefore, mitosis cell division takes place throughout your body. If you get hurt, if you are wounded, or any other problems that are occurring in your body, mitosis cell division takes place. Mitosis cell division takes place in two steps. The first one is karyokinesis and the second one is cytokinesis. Karyokinesis means nuclear division, where the nucleus divides itself, and cytokinesis is where the cytoplasm that is present inside the cell divides. In mitosis cell division, we have already done this part. Let us first revise what we have already done. In mitosis cell division, we learned about phases of division, right? So the first phase was prophase, second one was metaphase, third one was anaphase, and the fourth one was telophase. At the end of the fourth stage, we used to get two daughter cells and two independent daughter cells where the nucleus used to divide itself but also the cytoplasm used to divide. So, when the cytoplasm used to divide itself, there is where my cytokinesis takes place. Clear? Nuclear division takes place in telophase, but when cytoplasm divides itself into two different daughter cells, I call that process as cytokinesis process. So this is where mitosis cell division is completed. The same process or somewhat similar process is observed in meiosis cell division. Where are meiosis cell division taking place? It takes place in your reproductive organs. That is where the germ cells are present. Even meiosis cell division takes place in two stages, right? Directly we had prophase, anaphase, telophase, but over here we have two stages, meiosis 1 and meiosis 2. There are two stages of meiosis, meiosis 1 and meiosis 2. By meiosis, from one diploid cell, 4 haploid cells are formed. That means from one cell, how many daughter cells are formed? 4 daughter cells. Since 4 daughter cells are formed, we call it as haploid cells, right? And 2 cells, we call it as diploid cells. In meiosis, crossing over from non-sister chromatides of homologous chromosomes occur. That means crossing over takes place of sister chromatides and from there 4 daughter cells have been formed. This results in genetic recombination. The 4 daughter cells formed after completion of meiosis are genetically recombined and not exactly alike. The 4 daughter cells, they do not resemble each other. They genetically do not resemble each other and also not exactly similar to their parent cell. They even don't resemble to their parent cell. They may have some kind of similarities but not exactly how the parent cell looks like. Spores and gametes which are present in the germ cells. Spores and gametes are formed by meiosis. Meiosis helps to restore and maintain the chromosome number. That means from 2 chromosomes we have 4 chromosomes coming. Alright, in meiosis 1 we'll have the same process of karyokinesis 1 and cytokinesis 1. Similarly, in meiosis 2 also we would be having the same process, karyokinesis 2 and cytokinesis 3. In meiosis 1, all the stages would be prophase 1, metaphase 1, anaphase 1, telophase 1. But in prophase itself, we have 5 different stages. First is leptotene, second zygotene, third pachatine, fourth diplotin, fifth dikinases. Once this process is done, we have metaphase 1, we have anaphase 2, anaphase 1 and telophase 1. Again over here in telophase, how many processes we have? How many daughter cells? 2 daughter cells. That means cytoplasm gets divided into 2. These 2 daughter cells go ahead in meiosis. Again in telophase 1, right? These takes place once telophase 1 is completed, cytoplasms have been divided, 2 daughter cells have been given. Same process of meiosis 1, mitosis. Same process of mitosis takes place. How in mitosis you have prophase, metaphase, the same process takes place. But we'll be studying the 5 processes of prophase in detail in the next slide. But try to understand these things, that meiosis has 2 stages, meiosis 1 and meiosis 2. Since we have 4 daughter cells in the byproduct. Alright. So prophase 1 will be same as prophase in mitosis. Metaphase 1 will be same as metaphase in mitosis. Anaphase 1 will be same as anaphase in mitosis. Telophase will be same as telophase in mitosis. But over here in telophase we will be having 2 daughter cells. In meiosis, what is the byproduct? 4 daughter cells. So we require 2 more daughter cells. So that process takes place in meiosis 2. And at the end of telophase 2, the byproduct will be 4 daughter cells. Clear? And what is cytokinesis? Cytokinesis is where nuclear division takes place. Cytokinesis 1, we'll have 2 nucleus, 2 cells, daughter cells, and at the end of meiosis 2, cytokinesis will have 4 daughter cells. Okay. In karyokinesis 2 also we'll have prophase 2, metaphase 2, anaphase 2, telophase 2. And at the end of telophase 2, cytokinesis will take place and two cells will be divided into 4 cells, and the cytoplasm itself will also be divided. Now let us study 5 stages of prophase in detail. Prophase 1 of meiosis is much longer phase of meiosis. It is subdivided into 5 sub-stages, namely leptotene, zygotene, pachytene, diplotene and dikinases. Let us study leptotene in detail. Initially, the chromosomes start condensation and they become compact during leptotene. So what is happening? The chromatin fiber, whatever are they start becoming chromosomes. So once we get chromosome, they enter the second substage of zygotene. Zygotene means pairing, right? So these chromosomes which are formed, they start coming close by and standing parallel to each other. They start pairing. These chromosomes, they start pairing. In zygotene, homologous chromosomes start pairing. This pairing is called synapsis. The structure called synaptomenal complex develops to hold chromosomes in place during this pairing. Each chromosome's chromatide arm divides and forms structure like bivalent or tetrad. That means what is happening in zygotene, these chromosomes, they start pairing and stand close to one another. The next stage, once they are standing close to one another in pairs, they move on to the next stage, that is pachytene. In pachytene, during the pachytene stage, crossing over of non-sister chromatides, you can see they are crossing over each other. In the upper part, can you see the purple one and the pink one, they are crossing over each other. Chromatides of homologous chromosome takes place. Genetic recombination is produced due to such exchange. The homologous chromosomes still remain paired together at the site of crossing over. So, they are still paired amongst each other. The pair that was formed in zygotene stage, they are paired in pachytene stage and they start crossing over. Once they have crossed over, their chromatin fibers are exchanged among each other. They move on to the next stage, that is diplotene. During diplotene, synaptomenal complex dissolves and the homologous chromosomes of the bivalent separate except at the point of crossing over. Thus, it looks like X-shaped structure called the chismata. Chiasmata, right? So in diplotin stage, what is happening? They get separated from the centromere, they get separated and they are only joined on the part whichever they are crossed over each other, right? In the picture, as you can see, they got separated, but the part at which they have crossed over each other, that remains attached to each other in diplotin stage. The last stage, that is dikinesis, the last phase of prophase, is for termination of chiasmata. The spindle fibers originate and the crossing over homologous chromosomes are now separated. The nucleus disappears and the nuclear envelope breaks down. So what is happening? In dikinesis, they completely separate from each other. From the crossing over part also, they separate each other. And at this stage, the nucleolus dissolves, the nuclear membrane dissolves, and it's completely a part of the cell now. Then that particular cell enters into the metaphase 1 stage, then anaphase 1 stage, then telophase 1 stage. Once at telophase, we have two daughter cells. Cytoplasm gets separated and two daughter cells are produced. That is where cytokinesis 1 is completed. When cytokinesis 1 is completed, they immediately move on to meiosis 2. And again in meiosis 2, we have prophase 1. We don't have these 5 stages again. Prophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, and at the end of telophase 2, when we have 4 separate daughter cells, these 4 separate daughter cells will have separate cytoplasms, which will result to cytokinesis 2. And this is where cell division of meiosis is completed. So class, we have done the last part of the chapter, that is cell division by meiosis, meiosis cell division. You have to again view the video, read your chapter and revise. Write down the points which are being explained so that it's easier and it's simpler for you to understand what is mitosis cell division, what is meiosis cell division, and how does it take place. Thank you class. Stay home, stay safe, keep studying.